I have your test all the way scored, but probably by the end of school today, I will have all the tests in the grade book. For the ones that I've already graded and for my other two classes, I am so proud of you guys. I'm not worried about the younger generation like it seems like adults are. As those kids, they're going to ruin everything. The younger generations are, they've always gripe about. I was like, I'm not worried about you guys. Like you guys rock and roll. I'm worried about like my generation and the generation that's older. You guys are doing phenomenal. I have never seen test scores on average as high as what I've seen in this year in any unit. This one especially. I had very few D's and F's. And it usually like I would have like a lot of super high A's, hardly anything in the middle, and then lots of D's and F's on this unit. And you guys, like, you scored great. And even a lot of you, I could tell that you really did something different. I don't know what it was. I don't know what was different in this unit compared to last unit, but instead of scoring a D, you scored a B, or you jumped from a C to an A. So I just want to commend you because this is not an easy scenario. And you guys are doing better than what anyone is giving you credit for. Like, I'm so sick of our kids are going to be so far behind. Like, they're not going to, yeah, come to one of my classes and see what you guys are doing because you're doing great. Okay. So with that being said, I want you to grab your piece of paper and we're going to just draw a line down the center of it. I think photosynthesis is easier than respiration because respiration, we had to learn 4 steps like glycolysis, the prep reaction, okay? There's only 2 steps in photosynthesis, and one kind of looks like a review of oxidative phosphorylation, okay? And what can be, I think, so confusing, though, about photosynthesis is the fact that when they named these processes, they have like 5 different names for the second step. So you might be able to explain the second step, and that second step is called the Calvin cycle. But whenever they say, discuss the reduction of CO2, you're like, what does that mean? It's just the Calvin cycle. And you knew the Calvin cycle, but you didn't know that you can call the Calvin cycle that. And that's really what you were supposed to discuss, okay? So I'm going to kind of start over here with step number 2, because we know photosynthesis is all about making glucose. That's what's been hammered in your head. Photosynthesis makes glucose. But that glucose is actually made in the second step, okay? And what happens in that second step is carbon atoms from CO2 are used to build organic molecules. And lots of times we say like sugars are made in photosynthesis. We can say glucose is made in photosynthesis. We'll talk about some other things we can say is made in photosynthesis, okay? But this is just a general diagram. CO2 gets turned into sugars. And that's really what happens in the last step, okay? Now, as for the name of the second step, when I was younger, they like to call it, well, let me go back. This is what they call it now. They like to call the second step the light independent reaction. The light independent, and it's a series of reactions, like the Krebs cycle with several reactions. So is this step. It's several reactions. So that's why I put an S there. When I was younger, we just called it the light reaction or the light reactions. Okay. There was a guy with the last name of Calvin who discovered this series of reactions. And so he named it after himself. So I can also call it the Calvin cycle. And this brings back, think of the Krebs cycle, which you can also call the citric acid cycle. So this is the Calvin cycle, but we can also call it, oh, I'm really messed up. I'm sorry, guys. Did anyone notice my mistake? Let's come back over here. You got to erase. So if it's light independent, it means it doesn't need light. So, sorry, when I was growing up, the second step was called the dark reaction. But they stopped calling it really the dark reaction as much because it made students think that these reactions only happened in the dark. That is not true at all. Okay. So they kind of started getting away with calling it the dark reactions, but it's sometimes referred as that. Okay. So right now I have 3 names for this one step in photosynthesis. And we're going to add 2 more here in just a second, but they're not going to make sense until we diagram a few more things. All right. I'm going to draw another line here across the bottom of my page. I'm going to give me a few places to write here. So go ahead and do so. Okay. So let's think about what you guys already know. We know that if you, okay, first of all, sugars are CHO, right? If you want to take this molecule and turn it into a sugar, what element do you have to add to it? To go from here to here. What element? Hydrogen. Yes. Okay. So if we want to take CO2 and turn it into a sugar, we got to put H atoms on it. Okay. So for this to happen, this step needs hydrogen atoms. We need hydrogen atoms to put on these carbon atoms. So we can get carbon and hydrogen with those oxygens. Okay. And then the second main thing that the dark reaction or the light independent reaction needs is related to something you guys already know. I'm going to draw that graph again. Photosynthesis is an endergonic reaction. To take CO2 and basically convert CO2 with hydrogen atoms into glucose, we have to add energy because we are building a more complex, organized structure. We are decreasing entropy. So photosynthesis is an endergonic reaction, okay? And actually it's several reactions, several endergonic reactions. And you guys you know this. Anytime a cell needs energy for reactions to take place, what molecule can be used by cells to provide energy? ATP. Okay. So if a plant needs to take CO2, like 6 of them, and 12 hydrogens, and put them together, and that makes a sugar, by golly, that's going to take some energy to organize them, okay, and build a more complex structure, okay? All right. So let's go over here and let's talk about, those are the needs. Well, the first step is going to provide those 2 things that plants need to take CO2 and turn it into the sugar, okay? All right. So if this is called the light independent reaction, this one's called the light dependent reactions. The light dependent reactions. And what we're going to end up diagramming, these will not happen unless light is hitting the plant cell. Okay, so they're dependent on light. Okay. All right. We also sometimes just call it the light reaction, the light reactions. Okay, so sometimes we have the light reactions and then we have the dark reactions, or you say the light dependent reaction. So then they're going to call this step probably the light independent reaction. Okay. All right. I'm going to go down here kind of to this bottom square. Okay. So the first step supplies the H's. Okay. So this step supplies H's. Okay. And what this step's going to do is ultimately, remember H2O is needed. Remember, it's a reactant in photosynthesis. To simplify as much as possible, what happens is the H's get pulled off of the water and they get put on a bus. Do you remember the buses of respiration? Remember, NAD plus gets turned into NADH. FAD plus gets turned into FADH2 when you load those H's onto them, those protons and electrons, okay? The bus in photosynthesis is slightly different, but I think it's easy to remember because all you got to do is take NAD and put a P on it for photosynthesis. You know, that's really not what the P means, but to me, I'm like, okay, well, that's the photosynthesis one because it has a P in it for photosynthesis, okay? So in the light reaction, water's used. We pull the H's off of water, we stick them on the bus, and now it's NADPH. And I like to, remember the buses, I like to make them like bus color, okay? So this is the coenzyme. This is the proton and electron bus or shuttle. And just like the NADHs went somewhere and released their H's to the electron transport chain, the NAD pluses are going to grab those H's and take them where the dark reaction is occurring or where the light independent reaction is occurring, okay? Now, what it looks like is when you see like the Calvin cycle or the dark reaction, it's going to look like this. They're going to show you several steps because there's lots of intermediates. This gets turned into this, which gets turned into this, which gets turned into this. You don't have to have those memorized for my class, but they're going to show you the buses show up. Okay. So this is what we kind of diagram for the second step. NADPHs will bring the protons and the electrons and put them on these carbon atoms in CO2, basically. Okay, so let's review some terms. NADPH, is it getting oxidized or reduced?
Oxidized.
So that means the CO2 is getting
reduced.
That's a big deal. And the reason why I'm making a big deal about it is because sometimes if they say describe the reduction of CO2, it's this step. So sometimes they'll refer to this step as the reduction of CO2. So they're basically saying, describe how CO2 gets turned into a sugar, or describe the process or name the process where the CO2 is being turned into a sugar. I can say that it's being reduced, we're getting H's on it, and now we have a sugar, okay? So that's another way that they can refer to this. And like I said, students will know this pretty well, but when an essay question says, talk about the CO2 reduction in photosynthesis, they're like, but if I say describe the Calvin cycle, they can. So I just need you to know all the different ways they can refer to it, okay? All right. And let's then note down here in the corner, this step supplies the H's and gets them to the second step. It also supplies ATP. And kind of, again, show that ATP is chemical energy. It's an energy source that cells can use. Okay. All right. So we've kind of lied to you a little bit throughout your life when in your science classes. Because think of how many times you had a test question and the test question said, name the cell part that makes ATP. And you always said mitochondria, right? Okay. Well, we never told you this because we needed you to know that for state test. But technically you could be like, you could answer chloroplast and you would be correct because the chloroplast, through the light reaction, can actually make ATP. Now, usually to be a more accurate test question that students can't come back and argue that they were technically right on a state test, is the test question is going to say, name the cell part that makes ATP for the cell to use. Mitochondria makes ATP that can be used in the nucleus and in the cytoplasm for like activating kinases, all that stuff. I think of the chloroplast as kind of being like a narcissist. It makes ATP and it uses it all. It doesn't care about providing ATP, and it doesn't provide any ATP for any other parts of the cell. It makes it all, takes it all, uses it all, okay? All right. So when we look at, the light reaction or the Calvin cycle or the dark reaction, I don't care what you call it, just know it can be called multiple things, is lots of times, well, you're also going to see this. ATP is needed because I'll show you. We've got all these atoms. Here's CHs and Os. Okay, we have to turn these into one molecule of glucose. Think of how long it's going to take you to do that if I drop these on your desk. I'm like, okay, guys, here's your atoms. I got some H's for you and I got some CO2s. Build it into glucose. That takes ATP and phosphates that cause these atoms to be able to come together in different arrangements over several steps to equal this glucose molecule, okay? All right, so note that you're gonna see this happens. And it's not just once. You're gonna see other NADPHs because we're gonna need some here. They're in multiple steps, and we need some ATP here to be used. So just know you're gonna see NADPHs flying in, and you're gonna see ATPs flying in. And you need to be able to tell me why. The NADPHs are supplying the protons and electrons to reduce CO2 into the sugars. ATP provides energy so these atoms can be rearranged into a more complex structure. Okay? Let's see. Now, a few of the things, and I'm actually diagramming a lot on this side because when I was doing the original poster, and this one's not even done, this was so jumbled. I was like, man, they are not going to be able to see some of these main ideas I need them to know. So I'm doing some on one side, and then we'll actually go and see the processes and how these molecules get made here. Another thing that I didn't have much room to diagram is we got to talk about another lie that we said to you is, we always say CO2 gets turned into glucose, right? It doesn't. This molecule that is made in the light independent reaction is not glucose. Because glucose, remember, has 6 carbon atoms. This sugar that comes out only has 3 carbon atoms, and it's not glucose. It's called G3P. And you don't have to know the name. You don't have to remember G3P. They might talk about G3P. Again, that's just additional information in a paragraph that they're trying to kind of confuse you with on an AP question, okay? That molecule is called G3P. Now, they might refer to G3P as something else. By the way, so what is the prefix for 3? Because it has 3 carbon atoms. What's a prefix for 3?
Tri.
Okay. So G3P, they also might call it tri, but actually, what do we put on the end of sugars? What's the suffix for sugars?
Ose.
So does it make sense that G3P is a triose? It's a 3-carbon sugar, okay? Now, how many CO2s have to enter the dark reaction to get a triose? Do you need 3 carbon atoms? Well, CO2 only has 1. So that's why you're going to see on a lot of diagrams, it'll say 3 CO2s come in. That produces, and sometimes they won't show glucose coming out. They'll show it as a triose. But hey, when a cell, now let me talk about this G3P. Why G3P? G3P is very versatile. You can take it and you can turn it into amino acids, like our plant cells. because they have to make all of their organic molecules. They have to make their proteins, carbs, lipids, and nucleic acids. Plants don't run around eating things. Okay, they don't eat proteins, carbs, and lipids. They have to make their own. Okay, so they can turn it into amino acids. They can take G3P and they can turn it into fats. Because like when you eat pecans, you're eating oils in there. There's fats, vegetable oil and canola oil. So plants make their own fats too. Most of the G3P though this is what happens to it, is 2 of them are hooked together. So I put a ×2 there. And now we're finally to what we have been teaching you your whole lives. We just really try to simplify it, is cells can take 2 trioses and they combine them together. And this is finally glucose. Okay. All right. Now to the artwork. So either new piece of paper, or you can just flip over to the back here. I'm going to start with this a little bit further up and then I will zoom in. Okay. So I'm going to start with a little bit of blue sky. And the only reason why I'm really kind of zooming out is because I have seen so many questions where it says trace CO2 from where it enters a plant to eventually ends up as carbon atoms as glucose in a plant. So I'm trying to kind of really give you guys a big picture. So what I want to do is I want to start, and I'm going to make it a little bit, you can just do it light with some light blue here. I'm making like the sky. Okay. And this is a model, and a model always has some inaccuracies. I need to show you how plant cells are actually arranged into a leaf. Okay, so what I'm just going to draw here, I think what I'm going to do is open this up. Let me open this up and make it a little bit bigger here. Okay, so if you guys can look at my screen here, I'm going to draw, this is the plant cell that we're going to draw, okay? Up here is the outside, that's the air. Think of this as the blue sky, okay? This is the top of a leaf. We have plant cells. This is the bottom of the leaf, okay? And all these cells have chloroplast in them. All these cells can get, they have chloroplast and they can do photosynthesis, okay? Now, I need to make sure you understand what this is. Like I'm going to draw this cell, but next to this cell or next to this cell, I'm going to put these other little things, okay? This will make more sense in a second once we talk about this more, okay? I'm going to start off with drawing 2 little cells here. And I'm just going to start to label them. They're called guard cells. Okay. And the guard cells, they're actually pretty close together, but I'm just leaving a gap there. Together, so we have guard cells and then we have a hole where oxygen, sorry, actually I'm going to start with CO2. CO2 can go into our plant leaf. We will see oxygen come out of those holes out into the atmosphere, okay? So let me show you what guard cells and this opening is called the stomata. So this is called the stomata. It's the guard cells and an opening. Let me pull up a different view. Okay, so like on this one, you see, here's the 2 guard cells. They like flank the opening. They're like guards that stand beside a door and you can go through the door. Okay, so we have guard cells and then where we have the opening there, this whole structure is called the stomata. Okay, now let's look at what stomata look like if you look down on the top of a leaf. or you look at the bottom. They're really cool looking. This is exactly what they look like. So I'm looking at like the top of a leaf or the bottom of a leaf. So this is the surface of a leaf. This one has 1, 2, 3, 4, and this is an actual electron microscope image. This is a picture taken. This isn't a drawing. And look at all the holes that are all over a leaf. That's where the oxygen comes out, and that's where the CO2 goes in so that the cells nearby get the CO2 to do photosynthesis, okay? This is another image of what they look like, okay? And so if you look at this one, it's a little bit easier. This is one guard cell on this side, and then you have another guard cell on the other side. And what the guard cells can do, which we'll learn more about this here in the future, is the guard cells, when they're full of water, they look like jelly beans. Turgor pressure goes up, and it causes them to not be able to meet together, and there's a hole. The gas can go in and out of the plant cell. When a plant cell is, or plants are dry, like they haven't had rain, they're wilted, there's not a lot of water in the guard cells, and it's like a flat tire, and they can come together and they block the opening. One reason why they can open and close, the stomatas can open and close, is because not only can O2 and CO2 cross, but water can leak out through the leaves. It's called transpiration. And so if a plant is depleted in water, it cannot afford to lose any more. So the stomata closes. The guard cells come together and close it, and then the water can't escape. And then hopefully the plant can survive until it rains on just the water that it has, but it can't afford to lose any. So it closes up those holes. All right, so so we have guard cells, and then next to the guard cells, which I have to make this big, I'm going to put, and I'm not going to go all the way to the edge, so kind of look and see what I'm doing here. Okay, zoom up here. Ew. I'm drawing it more square, and that was just too ugly. I can't handle that. All right, too much of a perfectionist. All right, so plant cells are kind of more square, right? Okay, and really what the outside of a leaf is, it's just cell right next to cell. Like think of the cells that line your stomach. It's just one cell closely packed to another cell, and it's a barrier, okay? So this is the outside, and then we're in a cell in a leaf, okay? And then another cell's right next to it. There would be other cells over here. I can draw one if I wanted to, but okay. Because we lose perspective of where we're at, when you guys go back to study this diagram, I like to include other details, and I think it helps students figure out where on earth am I at, and where is this process actually happening in this plant and in a plant cell. So I want to go ahead and I want to put a cell wall in the cell membrane. So when you look at this, I think that double line might help you remember, okay, this is a plant cell. It's not the chloroplast. That's the plant cell. Okay, and also to give you some perspective here is I'm going to put, and this also helps us remember, I'm going to put a little tiny nucleus because they're eukaryotic, they have a nucleus. If I was really drawing kind of to scale, then the mitochondria, you know, this cell would be packed with mitochondria. So I'm just going to put a few mitochondria in here. Mitochondria look like circus peanuts. You know the circus peanuts that people eat? I always think that's what the mitochondria looks like. Okay, so I'm trying to help you guys remember that plants, yes, they have mitochondria. I'm going to zoom in here a little bit more. They have mitochondria that operate the exact same way as what we know. Their mitochondria does exactly, glycolysis happens, the prep reaction happens in the cell, everything. Okay, ATP is made there. Okay, and if I was going to draw a chloroplast, you know, chloroplasts are about the same size as mitochondria. And how we can tell that we have this circle as a chloroplast is because if you see the stacks of coins inside of them, you're looking at a chloroplast. Okay. Now, this is important because you know, usually when we draw a plant cell and I draw a big circle inside of a plant cell, usually what is that big circle? What is that big, huge circle that's usually in the middle of a plant cell?
Vacuole.
The vacuole, okay? I'm going to draw a big circle, but it's not going to be the vacuole. I'm going to draw a super huge chloroplast. And to help you realize that that circle is not a vacuole, that's a chloroplast, is I want you to do this. If you would grab green, and I want you to put just green around that little chloroplast that we drew. Also, what can you guys tell me about the membrane of chloroplast? One or two? Do you have a single membrane or a double membrane? Remember how endosymbiosis, mitochondria has two membranes, so does the chloroplast, because we think that their ancestors, or we know that their ancestors, because they have similarities to bacteria cells, they have two membranes, which means that they were engulfed by another cell, okay? All right, I'm going to put a little arrow and be like, okay, we are enlarge. We're going to enlarge this sucker and we're going to draw it really, really big, okay? And I mean, we're going to draw this, we're going to draw it big. So I'm going to draw a super huge chloroplast. And to help you realize this is not a vacuole, this is not a thylakoid, because I think we get lost a little bit in these diagrams because we have to make them look so different from what it would really look like. I'm going ahead and put two membranes around it. And since I colored, this chloroplast green here, I'm going to go ahead and make sure that with this green, I think when you guys are studying, you're going to realize, okay, that's a chloroplast. We just enlarged it. Okay. All right. The colors will help. Now, I've got to draw the coins inside of the chloroplast, but there's a problem. I can't really draw them to scale really small. I mean, I can. I'm going to put one like this. And so there's one coin. And I could do a stack, but I'm not going to. Just watch what I do and do something kind of like me, okay? So the thylakoids, so let's go ahead and label. So these would be these are thylakoids. And by the way, a stack, in case they refer to it, is called the grana. So if they say the grana, it's a stack of thylakoids. That's it. So if I have a stack of coins, each coin would be like the thylakoid. And I like to remember a thylacoin. So if you remember, a coin is one thylakoid, thylacoin, okay? So a stack is just simply called the grana. So I have two grana in this one. I'm using my terms. Okay, now here's the thing. I have to draw, like, there's a lot of action that happens in the thylakoid. So I'm going to draw a huge swollen thylakoid so we can actually diagram what's going to happen. So I'm going to draw a third thylakoid, but I'm going to make it big, but I'm not going to fill up this whole space. Probably should have. Okay. Okay. So let's make sure we know where we're at. Okay. So this is a chloroplast. And actually, that's the chloroplast double membrane. Okay. This would be the plant cell membrane. Okay, just to give you an idea where we're at. Plant cell membrane. Okay. So what would this be? What would I label this part?
Cytoplasm.
Cytoplasm. Okay. This thylakoid, this part is just a membrane. It's a phospholipid bilayer. It looks like every other membrane. So this is the thylakoid membrane. This is the thylakoid membrane. Now, I think we'll do some other things first before I label some more spots here. Let's start with, you guys know photosynthesis, chlorophyll, right? Chlorophyll is involved in photosynthesis. So let's kind of start there, right? Because light is absorbed by the pigment chlorophyll. Chlorophyll, as you all probably know, is green. So I want you to grab green. Now, chlorophyll and these other pigments are proteins. And proteins can be embedded in membranes, correct? Okay, so if this is a membrane, there's proteins embedded in it. So it kind of looks like what the cell membrane would look like with proteins embedded in it, okay? Now I'm gonna take my green, and in this location, I'm gonna draw, like they're embedded in the membrane, a few green circles. And I'm gonna zoom way in here and refocus. Okay, it's like I'm drawing grapes. And chlorophyll is not the only pigment that's in the thylakoid membrane. There's other pigments, and we know what color they are because we see them in the fall. We see carotenes in the fall, we see xanthophylline. They're orange. We see some pigments that are yellow. And so I'm just putting different colors in there to help you remember that it's just not chlorophyll that is in the membrane. It's not the only pigment. There's other pigments that can collect light energy, okay? Now, this cluster of pigments, like a grape cluster, this cluster of pigments has a name. And I'm gonna label it way up here. You gotta know that I'm labeling this, but I have too many other things I have to put here. This is called a photosystem. And I know it's the first one that I've drawn, but it's called photosystem 2. It's not photosystem 1. We're going to get to photosystem 1 here in a second. And a photosystem is a cluster of pigments in the thylakoid membrane. It's a cluster of pigments. So lots of times they won't draw it like this. They will just draw a blob, and that's the photosystem, okay? I'll show you, and you don't have to get out your other worksheet. Let me show you this. This is a photosystem. It's a bunch of pigments. And see how they try to show you as a bunch of pigments by kind of making the pigments as circles. So that's kind of why I did that, because I see that a lot on diagrams, okay? So this is photosystem 2, all right? There's other proteins embedded in the membrane, and we have drawn them before. We have drawn them in the inner membrane of the mitochondria. Remember them? We drew them like little circles. We said they were like the trampolines, right? Okay. Now, there's another photosystem. So I'm going to grab and I'm going to draw another photosystem, another cluster of pigments. So I'm just going to add some other colors to help remember there are more pigments besides chlorophyll. And different pigments are a good thing because different pigments can absorb different wavelengths of light. So a plant can get more out of sunlight if it has different pigments than just the chlorophyll. Okay, so again, this is a photosystem. And this one is called photosystem 1. We can abbreviate it PS. So this can be PS2, PS1. I am not talking PlayStation. Photosystem. Photosystem 1, photosystem 2. The reason why this one, it looks like it comes second, is named 1, and the one that comes first is named 2, is just the order in which they were discovered. So this cluster of pigments that's in this collective group of molecules that are going to work together here. It was discovered first. This one was discovered second. Okay, and then there's a couple more of those other proteins. And again, what did we call the proteins that were in the inner membrane of the mitochondria? Remember what they were called?
Electron, electron,
Transport chain. There you go. So here's an ETC, and there's then another electron transport chain, which is just a group of proteins. Okay, so we got some electron transport chain proteins embedded in the thylakoid. Okay, so let's start with kind of the first part of our equation. We know light, right? No light is needed for photosynthesis. Now let's talk about why or where. So light hits the pigments in the photosystem. So I'm showing light coming in. If you want to make it like a lightning bolt, go for it, okay? And when light hits the pigments, their electrons, that added energy makes those electrons excited, and they can leave those molecules. So we say light excites electrons, and the electrons from the photosystem can get passed to electron transport chain proteins. So this is like jumping from mattress to mattress, just like we did in the inner membrane of the mitochondria. And eventually they end up on this photosystem 1. Okay. And as light hits photosystem 2, it's also hitting photosystem 1. So let's show, light is hitting both photosystems. So we see the chlorophyll, the pigments, they're absorbing light. That light energy excites their electrons in the molecules of those pigments. And those electrons can get passed down the electron transport chain that's in the thylakoid membrane. Remember, we're inside of a chloroplast right now, looking at a coin. Now, I kind of skipped ahead here because as this electron gets passed to the first one, this is what's going to happen. Remember those H pluses? They can be moved across the electron transport chain. So every time an electron moves, protons are brought, and it's kind of different. Remember with photosynthesis or with respiration, remember the H pluses were moved out of the matrix? Now it's like you're bringing them in to the thylakoid. So the H pluses are being brought in and they're being concentrated. So if I could look at where it's more acidic, it's really acidic in the inside of a thylakoid because protons are being concentrated inside the thylakoid. Okay. And we all know what can happen if you put a bunch of H pluses, then they can flow on their own down an electrochemical gradient. And if the only thing they can flow through is ATP synthase, which is an enzyme that's also embedded in the thylakoid membrane. So we're moving from an area of, I'm going to write high concentration of protons to an area of low concentration of protons. This is coupled with a reaction, and that's the making of ATP. So ADP plus phosphorus gets turned into ATP. Okay. Finally, we have one molecule actually made that I said we needed to make. Remember, you got to make ATP to take CO2 and turn it into a sugar. So we finally got some ATP, right? Okay, now, just like in respiration, if the electrons can't move, this stops. You don't get the H pluses concentrated, you get no flow through ATP, so that you'll get no ATP. And what was the final, what came in and saved the day? What molecule can take that last electron off so that more electrons can be added and can flow? What was the final electron acceptor in respiration?
Oxygen.
Right. Well, we know that oxygen isn't needed in photosynthesis. There is a different molecule that is a final electron acceptor, and that is the NADP, a coenzyme, like NAD plus or FAD plus. Okay. It is the final electron acceptor. We always need something to take electrons off of the ETC, or the ETC can't keep adding electrons and passing them down. So it's like you can't jump onto the next trampoline if there's a kid on it already. So the parent has to come and grab the kid off the trampoline, and then you can jump from trampoline A to trampoline B because that kid got removed, okay? All right, so it pulls off the electron. And doesn't this make sense wherever we're at right here, there's H pluses, right? Because they're flowing. So there's H pluses here. They're highly concentrated in here though, but there's H pluses, all right? When the electron goes on to the ND plus, that negative pulls a proton with it, an H plus. And so we're always gonna show this. So NADP plus accepts the electron, binds to a proton or a hydrogen, and we just loaded the bus. Oops, NADP for photosynthesis, H. Okay. We made the bus. Okay. Now, let's see if this makes sense. Because by the way, I haven't talked about, like, we've got light. I haven't even mentioned yet and put on this poster. I haven't put water. I haven't even done the CO2 stuff yet. This is all the first step. This is the light reaction, okay? Let's talk about the water. Why plants must have water or this would shut down, photosynthesis would shut down, is if there was 10 electrons here, let's say that if we added up all the electrons, and I'm making this simple, if there's 10 electrons and they all got excited and left, this would be out of electrons, right? No more electrons can flow, no more H pluses can become concentrated, no more ATP can be made, okay? So that's where, I'm running out of space. I might have to change this and make a little bit of space here. Sorry, I need to make a little more space around here. Here is where the water is used and needed. So I'm gonna make a drop of water to help us remember where exactly the water is used. Okay. There is an enzyme that is activated by light. And I like to draw the enzyme because students sometimes tell me that light can split water, but that's not true. There's an enzyme that's activated by light and it binds to the water and it does this. It splits it. And it splits it into its pieces. It splits it into the H's. So if this is split, we would have H's come off. If there's an H plus, if an H comes off, then a proton comes off an electron, and look at what happens to the electrons. So the reason why photosystems never run out of electrons is because water is always donating electrons to the photosystems. So it never runs out. So the source of electrons to make ATP comes from water. Okay, and if you think about it, these H's that are all here came from the splitting of water. So these H's, and watch what happens. Remember H's, and then some of them get put on the bus. So ultimately, all of these H's came from water. Okay, so we'll use water to make ATP by providing electrons that can flow, concentrate, blah, blah, blah. Water also provides the H's, okay? All right, we will stop there. As for your homework assignment, There's only another lecture to watch. We have 4.6. I put it on Friday, but if you don't have 4.6 watched by Monday, it's not a big deal because I still need to talk about 4.5 stuff. So you can either watch 4.6 as homework today, or you can watch it like Monday as homework. It doesn't matter to me. Okay. It's whatever works best with your schedule. And remember, guys, if you need paper copies because you can't print off anything, then you can just come up to the high school, knock on my back door, and I will get you guys what you need. You might just send me an email, say, hey, I'm coming up, and I'll have things ready for you. Okay. All right. If you guys have any questions, feel free to stay on or jump on my meet later, and we'll see you guys later.
Hello, just curious, where is the excess oxygen go?
You watch him? So this oxygen that we end up making here, diffusion out into the air.
Okay, gotcha.
Some of it though, watch. Some of the oxygen will be used to make ATP.
Mhm.
Because remember, they do respiration to make ATP. So they're going to use some of the oxygen that they actually make. But there's always an excess of oxygen because photosynthesis happens at a faster rate than respiration does. So like if photosynthesis produces this much oxygen, then the respiration to make ATP would only use up like this much of it throughout the day. So there's always an excess of oxygen that can be released out of the plant cells from high concentrations, low concentrations out into the air, which then we can breathe it in.
All right, so that's why it's a product.
Yeah. Mm-hmm.
All right, gotcha. And also for the test I didn't take,
Okay.
I think I'm gonna do it sometime next week.
Okay.
I'll see how